Lecture Notes for AS Level Biology by Mohammed Rashid. Chat the cell number 030046104571. AS and a Level 9700 Smart Biology all in one. According to latest 2022-24 syllabus M. Rashid. Chat the 030046104571. Lecture Notes for AS Level Biology by Mohammed Rashid. Chat the cell number 030046104572. 1. Cell structure. All living organisms are made up of cells. New cells are formed by the division of pre-existing cells. Cells contain genetic material, which is passed from parent to daughter cells. All metabolic reactions take place inside cells. 1.1 The microscope in cell studies. The light microscope is an instrument used to magnify the specimen to 1,000 times its original size. In its simplest form, it is composed of a clear lens that magnifies the sample and a light source to illuminate it. Magnification can be calculated by using the formula. Comparing light and electron microscopes. Feature light microscope, electron microscope. Illumination. Light electrons focused by lenses, magnets. Maximum magnification x 1500 x 500 000. Resolving power / resolution 200 nanometers 0.2 m. 1 nanometer. Specimens living or dead. Preparation of specimens often simple. More complex. Cost of equipment relatively cheap. Very expensive. Images in color. Yes. Actual color of the specimen. No. Color is added by computer. Radiation used. Beams of light. Beams of electrons. Method of focusing. Glass lenses. Electromagnets. Maximum magnification of specimen being observed. Typical student microscopes magnify up to 400x the real size of the specimen. The maximum magnification of a research microscope is about 1500x. About 500,000x the real size of the specimen. Presence of vacuum within microscope. Absent scattering of light by air molecules does not affect image production. Present scattering of electrons by air molecules interferes with image production. Specimens that can be viewed. Specimens can be alive or dead. They can be stained so that different features can be distinguished. Specimens must be relatively thin so that light can pass through them. Specimens are always dead since they cannot survive inside a vacuum. Transmission microscope. Specimens must be extremely thin so that electrons can pass through them. Scanning microscope. Specimens need not be thin since beams of electrons are bounced off their surface. Lecture Notes for AS Level Biology by Mohammed Rashid. Chat the cell number 030046104573. 1.1 to 1 Temporary slide preparation. Temporary preparation of materials for light microscopy can be made for quick preliminary investigation. This may involve sectioning, staining and mounting. Fresh material may be hand-sectioned with a razor blade. A number of stains may be used for staining purpose, e.g. iodine solution, methylene blue, and eosin solution, etc. Biology specimens can be observed using either a light or electron microscope. There are two types of electron microscope, either a scanning or transmission electron microscope. Electrons are reflected from the specimen in a SEM and are passed through a TEM. Cell fractionation. Occasionally it is necessary to isolate organelles to investigate their structure or function, e.g. mitochondria could be used to investigate aerobic respiration away from the cell's internal environment. Cell fraction consists of two processes, homogenization followed by differential centrifugation. Cell fractionation depends on the different densities of the organelles. Technique. Cells kept at 5°C to slow down the autolysis, at isotonic solution to keep an ideal condition, and at a specific pH by a buffer solution. Homogenization is done by breaking cells into their components, cell fractionation. Centrifugation. separating these components using a centrifuge, ultracentrifugation. Cells homogenized and suspended in solution homogenate filtered to remove unbroken cells filtrate centrifuged and pellet of organelles removed if necessary, fluid supernatant can be spun again at a higher speed. New pellet is removed. Lecture notes for AS Level Biology by Mohammed Rashid Chat the cell number 030046104574. 1.1 to 2 Drawing cells from microscope slides and photomicrographs. Biological drawings are line pictures which show specific features that have been observed when the specimen was viewed. There are a number of rules/conventions that are followed when making a biological drawing. The conventions are: the drawing must have a title. The magnification under which the observations shown by the drawing are made must be recorded. A sharp HB pencil should be used, and a good eraser. Drawings should be on plain white paper. Lines should be clear, single lines, no thick shading. No shading is made. The drawing should take up as much of the space on the page as possible. Well-defined structures should be drawn. The drawing should be made with proper proportions. Label lines should not cross or have arrowheads and should connect directly to the part of the drawing being labeled. Label lines should be kept to one side of the drawing, in parallel to the top of the page, and drawn with a ruler. Drawings of cells are typically made when visualizing cells at a higher magnification power, whereas plan drawings are typically made of tissues viewed under lower magnifications. Individual cells are never drawn in a plan diagram. 1.1 to 3 Calculating magnification of images and actual sizes of specimens. Calculating size. Always measure in convert to real size using the magnification or the scale given. Use the most appropriate unit convert using the method. If you are given the magnification, you will often be required either to calculate the magnification of an object or to work out its actual size using a magnification given in the question. Basically, there are three things you have to know. The magnification, m, the size of the object that you can physically measure, the observed size, o, the actual size of the object, a. Lecture notes for AS Level Biology by Mohammed Rashid Chat the cell number 030046104575. 1.1 to 4 Use of eyepiece graticule and stage micrometer scale to make measurements. An eyepiece graticule, a little scale bar that you can place in the eyepiece of your light microscope. When you look down the microscope, you can see the graticule as well as the specimen. The graticule is marked off in graticule units, so you can use the graticule to measure the specimen you are viewing in these graticule units. Just turn the eyepiece so that the graticule scale lies over the object you want to measure. Calibration. The specimen is replaced with the stage micrometer. Focus on it using the same objective lens as you used for viewing the specimen. Line up the micrometer scale and the eyepiece graticule scale. You can do this by turning the eyepiece, and by moving the micrometer on the stage. Make sure that two large markings on each scale are exactly lined up with each other. Conversions. Biologists tend to measure small things, so you must be confident in using millimeters, mm. They have an easy-to-remember relationship and conversion is really straightforward. The units of measurement in biology are determined by SI, System International, units. The most commonly used include micrometers and nanometers. One meter, m, equals 1 x 10 om, 1 m, 1 decimeter, dm, equals 1 x 10 to 1 m, 1/10 m, 1 centimeter, cm, equals 1 x 10 to 2 m, 1/100 m, or 10 millimeters 1 millimeter, mm, equals 1 x 10 to 3 m, 1/1000 m, or 1000 micrometers 1 micrometer, μm, equals 1 x 10 to 6 m. 1/100000 m, or 1000 nanometers 1 nanometer, nm, equals 1 x 10 to 9 m. 1/10000000 m, or 10 o'clock p.m. 1 picometer, pm, equals 1 x 10 to 12 m. 1/100000000000 m. To convert one to the other. First decide whether the conversion is going to produce a larger or smaller figure. If you need to convert 50 millimeters to μm, remember 1 micrometer is smaller than 1 millimeter, so there will be a greater number of μm. But if you need to convert 50 nanometers to μm, remember that 1 micrometer is bigger than 1 nanometer. So there will be a smaller number of μm. Secondly, move the decimal point 3 places. Calculating size. Always measure in mm, convert to real size using the magnification or the scale given. Use the most appropriate unit, convert using the method. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 030046104576. If you are given the magnification, you will often be required either to calculate the magnification of an object or to work out its actual size using a magnification given in the question. Basically, there are 3 things you have to know. The magnification, m, the size of the object that you can physically measure, the observed size, o, the actual size of the object, a. These can be put into a simple equation: m equals o/a. Measuring cells. Cells and organelles can be measured with a microscope by means of an eyepiece called graticule. This is a transparent scale, usually having 100 divisions, figure A. The eyepiece graticule is placed in the microscope eyepiece so that it can be seen at the same time as the object to be measured, figure B. At this figure, figure B, the cell lies between 40 and 60 on the scale, so that it measures 20 eyepiece units in diameter. 60 to 40 equals 20. To calibrate the eyepiece graticule scale, a miniature transparent ruler called a stage micrometer scale is placed on the microscope stage and is brought into focus. This scale may be fixed onto a glass slide or printed on a transparent film. It commonly has subdivisions of 0.1 and 0.01 millimeters. The images of the 2 scales can then be superimposed, figure C. If in the eyepiece graticule, 100 units measure 0.25 millimeters, 0.25 x 1000 equals 250 more. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 030046104577. 1 unit equals 250 divided by 100 is equal to 2.5 its actual diameter equals 20 by 2.5 micrometers equals 50 micrometers. This diameter is greater than that of many human cells because the cell is a flattened epithelial cell. Use the following instructions to measure the length of one cell. Measure the distance in millimeter from the start of one cell to the end of 10 cells. Divide by 10 to find the length of one cell in the specimen. Convert this length in millimeter to micrometer by multiplying by 1000. Find the actual length of a cell by dividing this length by the magnification of the specimen got from the product of eyepiece and objective lens used. 1.1 to 5 resolution and magnification. Magnification is increasing the apparent size of the image, and resolution is the ability to distinguish between 2 points. Cells are the smallest unit of structure in organisms. Cells can be seen using microscopes. A microscope enlarges the image of cells. This is called magnification. To show us small structures within cells, a microscope must also be able to distinguish between objects that are close together. This is called resolution. An optical microscope passes light through a specimen to be viewed. Its maximum resolution is about 0.2 micrometers, 200 nanometers. An electron microscope uses beams of electrons. Its maximum magnification is about 500,000 times and its limit of resolution is about 1 nanometer. A transmission electron through a specimen to be viewed. A scanning electron microscope bounces beams of electrons off the surface of the specimen to be viewed. 1.2 Cells as the basic units of the living organisms. A plant cell as seen under electron microscope. 1.2 to 1 organelles of eukaryotic cells. Functions of major eukaryotic cells and structures. Cell organelles. Organelle occurrence size. Function cell surface membrane/plasma membrane. Encloses the cytoplasm of all cells. 7 to 10 nm thickness. Exchange and transport of materials into and out of the cell. Partially permeable. Prevents mixing. Lecture notes for AS Level Biology by Mohammad Rashid. Chat the cell number 030046104578. Nucleus usually 1 per cell. 10 m site of the nuclear material. The DNA which controls cell activities like protein synthesis and cell division etc. Nucleolus inside nucleus 1 to 2.5 m synthesizes ribosomes. Nuclear envelope around the nucleus 20 nanometers thickness bilayer membrane. The nuclear envelope, Ne, is a highly regulated membrane barrier that separates the nucleus from the cytoplasm in eukaryotic cells. It contains a large number of different proteins that have been implicated in chromatin organization and gene regulation. Rough endoplasmic reticulum. Continuous throughout cytoplasm. Extensive membrane network usually 0.2 m each cisterna. Synthesize proteins, isolate and transport newly synthesized proteins. Smooth endoplasmic reticulum. Usually small patches in cytoplasm. Variable, usually 0.2 m each cisterna. Synthesis of some lipids and steroids, cholesterol and detoxification. Golgi body free in cytoplasm 7 nanometers each cisterna. Modification and synthesis of chemicals e.g. glycoproteins, glycolipids. Mitochondria numerous in cytoplasm, up to 1000 per cell. 1 to 10 m aerobic respiration. Ribosomes free in cytoplasm or attached to rough ER. 18 or 22 nanometers, 70s, 80s, site of protein synthesis. Lysosomes free in cytoplasm 100 nanometers. Digestion of unwanted material with the help of hydrolytic enzymes, autolysis, phagocytosis, and autopsies. Centrioles small, hollow cylinders, containing a ring of microtubules, tiny protein cylinders. Centrioles/basal bodies are 0.2 to 0.5 micrometers in diameter. Involved with the separation of chromosomes during cell division, organize microtubules, spindle formation and help in cell division. Microtubule centrosome in animal cells 25 nanometers diameter. Microtubules are nucleated and organized by microtubule organizing centers, such as the centrosome found in the center of many animal cells or the basal bodies of cilia and flagella, or the spindle pole bodies found in most fungi. Cilia small, hair-like structures found on the surface membrane of some animal cells. In cross-section, they have an outer membrane and a ring of 9 pairs. There are 2 types of cilia, motile and primary, and they tend to be around 10 in length and around 0.2 m in diameter. The microtubules allow the cilia to move. This movement is used by the cell to move substances along the cell surface. Lecture notes for AS Level Biology by Muhammad Rashid. Chat the cell number 030046104579. Of protein microtubules inside, with a single pair of microtubules in the middle. Microvilli. These are folds in the cell membrane. The diameter ranges from 50 to 550 nanometers, length varies between 100 nanometers to several microns. They are found on cells involved in processes like absorption, such as epithelial cells in the small intestine. They increase the surface area of the cell membrane. Chloroplast. Cytoplasm of some plant cells, e.g. mesophyll. 4 to 10 m stroma traps light energy to convert chemical energy, e.g. synthesis of glucose and ATPs. Site of photosynthesis. Cell wall surrounds all plant cells, rigid, made up of cellulose. Plant cell walls vary from 0.1 to several m in thickness. Provides rigidity and strength and supports to plant cells, fully permeable, prevents the cell from bursting. Plasmodesmata. Plant cells 40 nanometers diameter. Plasmodesmata, PD, are gated plant cell wall channels that allow the trafficking of molecules between cells and play important roles during plant development and in the orchestration of cellular and systemic signaling responses during interactions of plants with the biotic and abiotic environment. Vacuole. Usually large, single fluid-filled space in plant, smaller and many in animals. Up to 90% of volume of whole plant cell. Storage of salts, sugars and pigments, creates turgor pressure by interaction with cell wall. Cytoplasm. Jelly-like substance found in all cells in between the cell membrane and nuclear membrane. Gelatinous liquid that fills the inside of a cell. It is composed of water, salts, and various organic molecules. Some intracellular organelles, such the nucleus and mitochondria, are enclosed by membranes that separate them from the cytoplasm. Cytoskeleton. Microtubules and actin filaments diameter of about 7 nanometers and are made up of many linked monomers of a protein. Shape of cell and movement of its parts. Flagellum. Flagella on eukaryotic cells are like cilia but longer. They stick out from the cell surface and are surrounded by the cell. 5 to 20 micrometers in length and 10 to 30 nanometers in diameter. The microtubules contract to make the flagella move. Flagella are used like outboard motors to propel cells forward, e.g. when a sperm cell swims. The flagella on prokaryotic cells have the same function, but a different internal structure. Lecture notes for AS Level Biology by Muhammad Rashid. Chat the cell number 03004610457010. Membrane. Inside they are like cilia, two microtubules in the center and 9 pairs around the edge. Microbodies. Membranous vesicles containing specific enzymes. 0.2 to 1.5 micrometers in diameter. Various metabolic tasks. Vesicle. A small fluid-filled sac in the cytoplasm, surrounded by a membrane. 3M. Transport substances in and out of the cell, via the cell surface membrane, and between organelles. Some are formed by the Golgi apparatus or the endoplasmic reticulum, while others are formed at the cell surface. Typical plant and animal cells. The major organelles found in eukaryotic cells with diagrams feature description function appears as 2 dark lines. These represent 2 layers of phospholipids. Separate cell from its surroundings controls movement of substances into and out of cell, allows cell identification and cell adhesion. Adjacent cell walls. Middle lamella layers of cellulose fibers around plant cells and of chitin around fungal cells absent from animal cells. Provides mechanical strength and support. Stops cell bursting in dilute solutions. Lecture notes for AS Level Biology by Muhammad Rashid. Chat the cell number 03004610457011. Chloroplast cigar-shaped organelle found in photosynthesizing cells. Its inner membrane has folds, called lamellae, which surround a fluid, called the stroma. Chlorophyll is found in the lamellae. Traps light energy and uses it to produce carbohydrates from carbon dioxide and water, i.e. photosynthesis. Endoplasmic reticulum (IR), ribosome. A series of tubes running through the cytoplasm. The tubes are surrounded by membrane. Rough ER has ribosomes studded into its membranes. Smooth ER has no ribosomes. Speeds up the distribution of substances through the cytoplasm. Golgi apparatus. A stack of flattened sacs surrounded by membrane. Small vesicles bud off its edge, not found in plant cells. Stores, and chemically modifies substances produced in the cell. These substances are later secreted using vesicles. Lysosome. A small sphere of liquid surrounded by membrane. The fluid contains powerful protein-digesting enzymes, lysozymes. These organelles are used to digest protein, e.g. they digest the cell after it dies. In some white blood cells, they are used to digest bacteria. Microvilli in plasma membrane. Microscopic folds in the plasma membrane only found in some types of epithelial cells. Increases surface area of membrane for absorption. Lecture notes for AS Level Biology by Mohammad Rashid. Chat the cell number 03004610455712. Mitochondrion. Cigar-shaped organelle found in all cells. Its inner membrane has folds, called cristae, which surround a fluid matrix. Produces adenosine triphosphate, ADP, which cells use as an energy source. This is the process of respiration. Nucleus, nuclear pore. Nucleolus contains chromosomes and stores of nucleic acid, in one nucleolus or in several nucleoli, separated from cytoplasm by a nuclear envelope made of 2 layers of plasma membrane containing small holes, called nuclear pores. Chromosomes contain genes made of deoxyribonucleic acid, DNA. These genes control the cell's activities. This can be seen only as a small black sphere. Sometimes ribosomes are free in the cytoplasm, sometimes they are bound to ER, forming rough ER. Using chemical instructions from the nucleus, ribosomes assemble amino acids to make proteins. 1.2 to 2 Typical plant and animal cells. Lecture notes for AS Level Biology by Mohammad Rashid. Chat the cell number 03004610455713. 1.2 to 3 Structure of typical plant and animal cells. Comparison of prokaryotic cell, plant cells and animal cells. Feature prokaryotic cell, eukaryote animal cell. Plant cell 70s ribosomes. Yes, free in the cytoplasm. Yes, some free and some are attached with RR. Yes. ADS ribosomes. No. Yes. Yes. Capsule. Yes. No. No. Cell wall. Yes. Murium. No. Yes. Centrioles. No. Yes. No. Cytoplasm. Yes. 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 Cytoskeleton. No. Yes. Yes. Golgi bodies. No. Yes. Yes. Lysosomes. No. Yes. Yes. Mesosome. Yes. No. No. Mitochondria. No. Yes. Yes. Nucleus. No. Yes. Yes. Plasma membrane. Yes. 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 Plastids, e.g. chloroplasts. No. No. Yes. Rough ER. No. Yes. Yes. Smooth ER. No. Yes. Yes. Vacuoles. No. No. Yes. Vesicles, small spheres of liquid surrounded by membrane. No. Yes. Yes. Lecture notes for AS Level Biology by Mohammad Rashid. Chat the cell number 03004610455714. Difference between prokaryotes and eukaryotes. Eukaryotes history 3.5 billion years ago 1.5 billion years ago organisms bacteria plants, animals, protoctists. Fungi nucleus no true nucleus or nuclear membrane nucleus present with nuclear membrane diameter of cells 0.5 to 10 m 10 to 100 m site of genetic material DNA in cytoplasm DNA inside distinct nucleus organization of genetic material DNA is circular. Plasmid, no histone proteins. DNA does not condense at cell division. DNA is linear, attached to histone proteins, condenses into visible chromosomes before cell division. Internal structure few organelles, small ribosomes, 70s, no chloroplast, only bacterial chlorophyll associated with cell surface membrane called mesosome, no endoplasmic reticulum or associated Golgi body in lysosomes. Many organelles with double membrane system, mitochondria, chloroplasts, nucleus, some have single membrane, lysosomes, Golgi apparatus, endoplasmic reticulum, rough and smooth, vacuoles some have no membrane, ribosomes, centrioles and microtubules nitrogen fixation some to make amino acids none cell wall always present but made up of peptidoglycan, murine. Present in plants made up of cellulose and fungi made up of chitin and some protoctists, never. Animals' flagella have simple flagella without microtubules have modified cilia that consist of microtubules in a distinctive 9+2 arrangement cell division no mitosis or meiosis occurs but by binary fission without involving a spindle. Mitosis and meiosis occurs and involves a spindle. Lecture notes for AS Level Biology by Muhammad Rashid Chat the cell number 03004610455715. Protein synthesis protein synthesized in small ribosomes protein synthesized in large ribosomes. Organisms can be classified as viruses, prokaryotes, and eukaryotes. Viruses prokaryotes eukaryotes example flu virus, HIV bacteria plants, animals, fungi, protoctist size 10 to 30 nanometers, 0.1 to 10 micrometers, 10 to 100 micrometers, nuclear material either RNA or DNA plus proteins. Circular DNA, no histone proteins. Not organized within a membrane-bound nucleus. Linear DNA attached to histone proteins. Organized within a membrane-bound nucleus. Internal organization no membranes or organelles no membranes or organelles complex membranes compartmentalize the cell. Structures normally visible in animal and plant cells with the light microscope organelle function animal cells plant cells nucleus control of cell activities yes storage and support, its membrane is called tonoplast no yes. Chloroplasts photosynthesis no yes cell wall support, protects and gives shape to cell no yes. Plasma membrane barrier controlling entry to and exit from cell yes yes. Nuclear envelope / membrane contains the nucleus, the pores for movement of materials yes yes. DNA heredity material yes yes mitochondria site of part of respiration yes yes endoplasmic reticulum synthesis and movement of substances in the cytoplasm yes yes. Ribosome site of protein synthesis yes yes Golgi apparatus site of synthesis of cell secretions yes yes. Lysosomes contains lytic enzymes always often microbodies usually cytoskeleton yes yes centrioles beside the nucleus, animals, yes no, in large plants, 9+2 cilia or flagella locomotion, current creation, feeding often no, in flowering plants, yes, in ferns, cycads and bryophytes. Lecture notes for AS Level Biology by Muhammad Rashid Chat the cell number 03004610455716. 1.2 to 4 Use of ATP by cells from respiration. Active transport is a process by which cells move molecules or ions against their concentration gradient. It involves the use of energy by cells. Like facilitated diffusion, this process also involves carrier proteins in membranes. Endocytosis is bulk movement of substances into a cell. In this case, membranes become trapped in a vesicle that buds off from the plasma membrane. In phagocytosis, the trapped exocytosis is the bulk movement of substances out of a cell. These substances become trapped inside a vesicle formed in the cytoplasm. They are secreted when the vesicle moves to the plasma membrane and fuses with it. 1.2 to 5 key structural features of prokaryotic cell. Prokaryotic cells do not have a nucleus that is separated from the cytoplasm by a nuclear envelope. Organisms other than bacteria have eukaryotic cells. Prokaryotic cells are surrounded by a cell wall and a plasma membrane. Some are surrounded by a capsule. Ribosomes are the only organelles found in their cytoplasm, 70s ribosomes only. Cells have at least one chromosome that controls its activities. Prokaryotic cells have a single, circular chromosome that is in their cytoplasm. Eukaryotic cells have several linear chromosomes that are found in the nucleus. 1.2 to 6 Comparison of prokaryotic cells with eukaryotic cells. Comparison of prokaryotic cell, plant cells and animal cells. Feature prokaryotic cell, eukaryote animal cell, plant cell. 70s ribosomes, yes, free in the cytoplasm, yes, some free and some are attached with RR. Yes. 80s ribosomes, no, yes, yes, capsule, yes, no, no, cell wall, yes, murion, no, yes, made up of cellulose in plants and chitin in fungi. Centrioles, no, yes, no, cytoplasm, yes, 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 cytoskeleton, no, yes, yes, Golgi bodies, no, yes, yes, lysosomes, no, yes, yes, mesosome, yes, no, no, mitochondria, no, yes, yes, nucleus, no, yes, yes, nuclear envelope, no, yes, yes, nucleoli, no, yes, yes, chromosome, single and circular, many and linear plasma membrane, yes, 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 plastids, e.g. chloroplasts, no, no, yes. Rough ER, no, yes, yes, smooth ER, no, yes, yes, vacuoles, no, no, yes. Lecture notes for AS Level Biology by Mohammed Rashid Chat. The cell number 03004610457717. Vesicles, small spheres of liquid surrounded by membrane. No, yes, yes. 1.2 to 7 Viruses as non-cellular structures. Viruses are disease-causing agents, rather than organisms. The distinctive features of viruses are, they are not cellular structures, but rather consist of a core of nucleic acid, DNA or RNA, surrounded by a protein coat, called a capsid. In some viruses there is an additional external envelope or membrane made of lipids and proteins, e.g. HIV. They are extremely small when compared with bacteria. Most viruses are in a size range of 20 to 400 nanometers, 0.02 to 0.4 micrometers. They become visible only by means of the electron microscope. They can reproduce only inside specific living cells, so viruses function as endoparasites in their host organism. They have to be transported in some way between host cells. Viruses are highly specific to particular host species, some to plant species, some to animal species and some to bacteria. Viruses are classified by the type of nucleic acid they contain. Key words: cell, nucleus, organelles, eukaryote, prokaryote, cell surface membrane, chromatin, chromosomes, nucleolus, cytoplasm, protoplasm, mitochondria, units of measurement, centrioles and centrosomes, cell wall, plasmodesmata, singular plasmodesma, vacuoles, tonoplast, chloroplast, photosynthesis, grana, singular granum, magnification, resolution, actual image. M equals I/A, light microscope, electron microscope, stage micrometer, eyepiece graticule, real size of an object, electro. Magnetic spectrum, transmission, FTM, and scanning, SEM, electron microscope, viewing specimen with the electron microscope, microvilli, lysosomes, endocytosis, exocytosis, self-digestion, ATP, Christie, ADP, endosymbiont theory, microtubules and microtubules organizing centers, tox, cilia, cilium, flagella, flagellum, thylakoids, bacteria, virus, mesosome, peptidoglycan, plasmid, pili, pilus, phospholipids, adenosine triphosphate, ATP, capsid. Eukaryotic cell, Golgi apparatus / Golgi body / Golgi complex, microvilli, nuclear envelope. Photomicrograph, prokaryotic cell, rough endoplasmic reticulum, RR, smooth endoplasmic reticulum, SER, viral envelope, capsomere. Chapter Glossary. ATP is a nucleotide derivative which acts as the energy currency in cells. It consists of a molecule of ribose joined to the nitrogenous base adenine and 3 phosphate groups. Capsid is the outer protein shell of a virus that encases and protects the viral genome. Lecture notes for AS Level Biology by Mohammad Rashid Chat. The cell number 03004610457718. Capsomere is the envelope that protects the viral genome and thus helps the virus to escape from recognition by the host immune system. Cell surface membrane is a phospholipid bilayer studded with proteins that surround cells and separates them from their environment. Cell wall is a permeable layer made of polysaccharides that surrounds plant, algal and fungal cells. Centrioles are cytoplasmic structures made of microtubules that produce the spindle fibers during mitosis. Chloroplast is an organelle found in plants and algae that is the site of photosynthesis. Cilia are small, hair-like organelles that extend from the surface of eukaryotic cells. They have modal and sensory functions. Electron micrograph is an image of a non-living structure produced by a scanning electron microscope, SEM, or a transmission electron microscope, TEM. Eukaryotic cell is a type of cell that contains a nucleus along with membrane-bound organelles. Eyepiece graticule is a scale bar inside the eyepiece of a light microscope which can be calibrated against a ruler to measure structures. Golgi apparatus / Golgi body / Golgi complex is an organelle found in eukaryotic cells that is involved in the modification and packaging of proteins. Lysosomes are membrane-bound vesicles in the cytoplasm that contain a hydrolytic enzyme called lysozyme. Magnification means how much bigger an image appears compared to the original object, calculated using the following formula. Microvilli are microscopic finger-like projections from the plasma membrane of some animal cells. Mitochondrion, PL mitochondria, is an organelle found in eukaryotic cells that is the site of aerobic respiration. Nuclear envelope is a double membrane that surrounds the nucleus. Nucleolus is a structure found inside the nucleus that contains proteins and RNA and is involved in synthesizing new ribosomes. Nucleus is an organelle found in eukaryotic cells that stores the genetic information of the cell as chromosomes and is surrounded by a membrane called the nuclear envelope. Peptidoglycan is a polymer consisting of amino acids and sugars that forms a 3D mesh and makes up the cell walls of most bacteria. Photomicrograph is an image produced from a light microscope, also known as an optical microscope. Plasmodesmata are microscopic channels between plant cell walls that facilitate symplastic transport. Prokaryotic cell is a type of cell that does not contain any membrane-bound organelles or a nucleus. Resolution is the ability to distinguish two different points in a specimen. Ribosomes are organelles found either free in the cytoplasm or membrane-bound that are involved in the synthesis of proteins. Rough endoplasmic reticulum, RR, is a membrane-bound organelle that is involved in the synthesis and packaging of proteins. Lecture notes for AS Level Biology by Mohammad Rashid Chat. The cell number 03004610457719. Scanning electron microscope, SEM, is a type of electron microscope that passes a beam of electrons over the surface of a specimen to produce an image. Smooth endoplasmic reticulum, SER, is a membrane-bound organelle involved in lipid synthesis. Stage micrometer is a scale that may be mounted to the stage of a light microscope and can be used to calibrate an eyepiece graticule. Tonoplast is the lipid bilayer that surrounds a permanent vacuole. Transmission electron microscope, TM, is a type of electron microscope that passes a beam of electrons through a sample to produce an image. Vacuole, permanent, is a membrane-bound structure found in plant cells that contain cell sap. Virus is a non-living microorganism that consists of genetic material surrounded by a protein husk. Viral envelope is the outermost layer of many types of viruses that consists of phospholipids. Short MCQs with right answers. 1. Freeze fracture technique of onion root cells organelles, nucleus, where structure X represents. The cells were broken apart at the nuclear envelope, structure X is a nuclear pore. 2. Which cell structures can form vesicles? 3. Which cells contain a tonoplast? 4. Which organelles found in animal or plant cells are surrounded by double membranes? Chloroplasts, mitochondria, nuclei 5. Some scientists think that mitochondria evolved from bacteria that entered the cytoplasm of a different cell and were able to survive there. Which structural features of mitochondria support this hypothesis? 6. What is present in all viruses, all prokaryotes, and all eukaryotes? Cytosine 7. A graticule and a micrometer scale can be used to measure the size of biological structures that are viewed with a microscope. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455720. Which row shows the correct locations for the placement of a graticule and a micrometer scale on the microscope shown? 8. Which organelles are involved in the synthesis and secretion of a glycoprotein? 9. Which cell structures can have mRNA inside them? 1. Chloroplast 2. Mitochondrion 3. Nucleus 10. A scientist carried out an experiment to separate the organelles in an animal cell by mass. The scientist mixed the cells with a buffer solution which had the same water potential as the cells. The cells were broken open with a blender to release the organelles. The extracted mixture was filtered and then spun in a centrifuge at a high speed to separate the heaviest type of organelle. These sank to the bottom, forming solid pellet 1. The liquid above pellet 1 was poured into a clean centrifuge tube and spun in the centrifuge at a higher speed to separate the next heaviest type of organelle. These organelles sank to the bottom, forming solid pellet 2. This procedure was repeated twice more to obtain pellet 3 and pellet 4, each containing a single type of organelle. What is the main function of the type of organelle extracted in pellet 2? Production of ATP. 11. Which structures are found in palisade mesophyll cells and photosynthetic prokaryotes? 1. Cell surface membrane. 2. Ribosomes. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455721. 12. Which polymers are present in all viruses and all prokaryotes? 1. Polynucleotides. 2. Polypeptides. 13. Which feature is visible with a light microscope using a natural light source? Paramecium cell of diameter 200 m14. The electron micrograph shows a structure found in the cytoplasm of an animal cell. Which biological molecules are found in this structure? Proteins. Which cell structures contain nucleic acid? 1. Cytoplasm. 2. Mitochondria. 15. Mitochondria are thought to have evolved from prokaryotic cells that were ingested by an ancestral cell. Which feature have prokaryotes lost during their evolution into mitochondria? Cell wall 16. Which polymers are present in all viruses, all prokaryotes, and all eukaryotes? 1. Polynucleotides. 2. Polypeptide 17. How many of the listed structures typically contain genetic material that has telomeres? Nucleus. 1, 18. The photomicrograph shows cells from a human blood smear. Which calculation shows a correct method to calculate the actual diameter of the neutrophil shown in the photomicrograph in micrometers? M. 19. A student calibrated an eyepiece graticule using a stage micrometer. Each division of the stage micrometer was 0.01 millimeters. With AX10 magnification objective lens, 10 eyepiece graticule units match 10 divisions on the stage micrometer. The same microscope was used with AX40, instead of AX10, magnification objective lens to measure the diameter of an alveolus. The diameter of the alveolus was found to be 96 eyepiece graticule. Lecture notes for AS Level Biology by Mohammed Rashid Chat. The cell number 03004610457722. Units. The eyepiece lens was not changed. What is the best estimate for the diameter of the alveolus? 240 m. 20. Which statements are correct for chloroplasts and also for mitochondria? 1. They can transcribe their circular DNA. 2. They can translate mRNA. 3. They are enclosed by double membranes. 21. The electron micrograph shows some cells from a root. Which cell structure is not usually found in cells from a root? B. Chloroplast. 22. A scale bar on an electron micrograph is 2 centimeters long and represents an actual length of 1 micrometers. What is the magnification of the electron micrograph? X 20,023. The mean width of mitochondria in an electron micrograph is 6 millimeters. The magnification of the electron micrograph is X 9600. What is the actual mean width of the mitochondria? 6x10-4 mm24. The actual diameter of a prokaryotic cell is 0.5 micrometers. An electron micrograph of the cell has a magnification of x 50,000. What is the diameter of the cell in the image? I equals mx a equals 0.05 x 50,000, 2.5 by 101 millimeters 25. What is the definition of the resolution of a light microscope? The minimum distance that allows two objects to be viewed as separate. 26. How many nanometers are there in 1 millimeter? 1 times 1000 times 1000 is equal to 1,027,000. What is the typical resolution of a microscope using daylight as a light source with AX10? Eyepiece lens and AX40 objective lens. 200 nanometers. 28. A prokaryote cell, which is 1 micrometer in diameter, is magnified 50,000 times in an electron micrograph. What is the diameter of the cell in the electron micrograph? 5 by 101 millimeters 29. Which equation for calculating the actual size of a specimen, A, or image size, I, or magnification, M, is correct? M equals I divided by A30. Which definition of the magnification of a drawing of a leaf is correct? The size of the drawing of a specimen in comparison to the actual size. 31. A light microscope is used to observe two structures that are 200 nanometers apart. How far apart are the structures when the magnification is changed from x40 to x400? 200 nanometers. 32. Which calculation is used to find the actual length of an organelle from an image? Image size divided by magnification. 33. A student was presented with a photomicrograph of a cell organelle. The magnification of the photomicrograph is known. Which calculation of the actual length of the organelle in Mu-M is correct. Image size in mmx 1000 divided by the magnification. 34. A student has drawn a cell structure as seen using a light microscope. The magnification of the drawing is x 600. The length of the structure on the drawing is 6 mm. What is the actual length of the cell structure? A equals I divided by m, 6 mm equals 6000 micrometers, 1 by 101 micrometers 35. Red blood cells have a diameter of 7000 nanometers. Pancreatic cells have a diameter of 35 micrometers. What is correct about the relative sizes of these cells? 35 micrometers equals 35,000 nanometers. The red blood cells are 5 times smaller. Lecture notes for AS Level Biology by Muhammad Rashid Chat. The cell number 03004610455723. 36. When making measurements in experiment, which methods could have parallax errors? 1. Using a calibrated eyepiece graticule to measure length. 2. Using a measuring cylinder to measure volume. 3. Using a ruler to measure length of a shoot. 2 and 3 only 37. At approximately which magnification is light microscopy not suitable because the resolution becomes too low? X 150 38. What best describes an electron microscope in comparison with a light microscope? Higher magnification and higher resolution. 39. What is not a limitation of an electron microscope? There is an increase in resolution and magnification compared with the light microscope. 40. Which structure is measured in the units shown using a light microscope? Chloroplast mu-M 41. Which statement is not correct in its description of a light microscope or an electron microscope? An electron microscope has a maximum resolution of 0.05 nanometers. 42. An electron microscope has a higher resolution than a light microscope. Which is a result of the higher resolution? The ability to see crystal in mitochondria. 43. A light microscope is used to observe two membranes that are 200 nanometers apart. How far apart are the membranes when the objective lens is changed from low power, x40, to high power, x400? 200 nanometers 44. Which eyepiece and objective lens combination of a light microscope enables you to see the greatest number of cells in the field of view? X5 eyepiece lens and X10 objective lens 45. Which statement about the light microscope is correct? If the resolution is 220 nanometers, then a bacterium 0.2 micrometers, 200 nanometers, in diameter will not be visible. 46. What restricts the resolution of the light microscope? The wavelengths of visible light. 47. Which range of sizes would include most eukaryotic cells? 1 by 101 micrometers to 1 by 102 micrometers 48. An eyepiece graticule can be calibrated using a stage microscope. What is the correct reason why an eyepiece graticule is calibrated? An eyepiece graticule can be used to make measurements. 49. Which group of structures are visible in a suitably stained plant cell using a high power? X400, light microscope. Centriole, not visible. Chromosomes, not visible. Mitochondria, visible. Starch grains, visible 50. Which eukaryotic cell structures all contain nucleic acids? Chloroplasts, mitochondria, cytoplasm, ribosomes. 51. Microvilli and root hairs are characteristic structures of some cell types. Which row identifies some features of a microvillus and a root hair? Surrounded by cell wall, root hair, more than one present on a cell, microvillus 52. Which cell organelles are clearly visible when viewed with a light microscope at high power, X400? Ribosomes, no. Endoplasmic reticulum, no. Centrioles, no. Chloroplasts, yes 53. Which cell structures contain rRNA? chloroplast, mitochondrion, and nucleus 54. Which cell structures are required for the formation of lysosomes and the hydrolytic enzymes that they contain? Mitochondria, rough endoplasmic reticulum and Golgi body. 55. In which cell structures does DNA transcription occur? Chloroplast, mitochondrion. Nucleus 56. Which row is correct for structures found in eukaryotic cells? Circular DNA, present, 70s ribosomes, present, 80s ribosomes, present 57. Which cell structure contains DNA? Mitochondria, chloroplasts, and nucleolus 58. Which sequence shows the correct order of some of the stages in the production and secretion of an enzyme? Ribosome, rough endoplasmic reticulum, vesicle, Golgi body 59. Which row describes the membrane surrounding each cell structure? Chloroplast, double, mitochondrion, double, nucleus, double 60. Which cell structures contain nucleic acid? Mitochondria and ribosomes. Lecture notes for AS Level Biology by Mohammed Rashid. Chat to cell number 03004610455724. 61. Which parts of cell contain ribosomes? Chloroplast, mitochondrion, and cytoplasm 62. Which part of cell is often continuous with the rough endoplasmic reticulum? Nuclear envelope 63. Insulin is a hormone synthesized from two polypeptide chains inside cells of the pancreas. Which row shows the cell structures that are used in producing insulin and releasing it from the cells? Golgi body, used, lysosome, not used, rough endoplasmic reticulum, used 64. Which row shows the correct order of size of these cell structures? Large, small, width of a chloroplast, width of a mitochondrion, width of a ribosome, width of a cell surface membrane 65. Some stains can be used to identify cell structures in living cells. A dilute solution of one stain causes the whole cell to appear blue. The blue color rapidly disappears in most cell structures. Those cell structures that release energy stay blue. Which type of cell structure is likely to stay blue? Mitochondrion. 66. Which row identifies the type of ribosome found in each of the different structures? Chloroplast, 70s, cytoplasm of eukaryotic cell 80s, prokaryotic cell, 70s, mitochondrion, 70s 67. The electron micrograph shows part of a eukaryotic cell. Which cell structure is a site of protein synthesis? Ribosome 68. Which row correctly shows the features of a cell structure? Site of protein synthesis, ribosomes, makes lysosomes, Golgi body 69. Which structures are found in typical eukaryotic cells? 70s ribosomes, 80s ribosomes, linear DNA, chromosomes, and circular DNA 70. In a photomicrograph, which structure shows that it is a plant cell? It has chloroplast. 71. Which cell structures contain RNA? Mitochondria, nucleus, and ribosome 72. Which cell structures can form vesicles? Cell surface membrane, ER, and Golgi body 73. Plant cells are stained and then seen with a simple light microscope using daylight as the only light source. Which cell structures are clearly visible at a magnification of x 400? Nucleoli. 74. Which cell structure contains 70s ribosomes? Chloroplast and mitochondria. 75. Which structures are found in animal cells and in plant cells? Lysosome, nucleolus, and vacuole 76. Which cell structure contains cytoplasm? Plasmodesmata 77. Some cell structures are listed in a particular order. 1. Nucleus 2. Ribosomes 3. Golgi body 4. Vesicle. What determines the order in which these cell structures are listed? Sequence used in synthesis of antibody. 78. Lysosomes vary in shape and size, making them difficult to identify. What describes a lysosome? A vesicle enclosed by a single membrane, containing several different hydrolytic enzymes that may act inside or outside the cell. 79. Which features of cilia and root hair cells are correct? Increases cell surface area. Root hairs cannot be resolved with the light microscope. Cilia contain vacuoles, root hairs, more than one present on a cell. Cilia. 80. Which are functions of microtubules? Allowing movement of cilia in a bronchus. Attachment of centromeres during metaphase and moving secretory vesicles. Around a cell. 81. What is the correct order of cell structures from largest to smallest? Nucleus, chloroplasts, mitochondria, ribosomes. 82. Which cell structures have ribosomal RNA? Chloroplast, mitochondrion, nucleus and rough endoplasmic reticulum. 83. What is the function of rough endoplasmic reticulum, RER, in an animal cell? Protein synthesis and transport. 84. Where would nucleic acid be found in a typical animal cell? Cytoplasm, RER, and mitochondrion. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455725. 85. Which statement explains why it is necessary to use an electron microscope to see the cristae of a mitochondrion? The resolution of a student microscope using daylight is too low. 86. The removal of a plant cell whale by enzyme digestion leaves an intact membrane-bound structure called a protoplast. Which statement explains why protoplasts are easily damaged? Net movement of water is no longer limited by turgor. 87. Which structures are present in a Vibrio cholerae cell? Circular DNA. 88. Which cell structures may contain cisternae? Endoplasmic reticulum and Golgi body. 89. Which row correctly describes the function of the cell structures? Lysosomes, digestion of unwanted structures, mitochondria, ATP synthesis, smooth endoplasmic reticulum, lipid production, Golgi body, glycoprotein production. 90. The diagram shows the relationship between various cells and their components. Which row is correct? 80S ribosome, eukaryotic cell, mitochondrion, 70S ribosome. Prokaryotic cell. 91. Which features can be found in plant cells and in prokaryotic cells? Cell wall, cell surface membrane and ribosomes. 92. Which type of cell contains the highest proportion of cell structures found by a single membrane? Golgi body. 93. Which row shows the structures that could be found in the root cells of a plant? Plasmodesmata, Golgi body, and mitochondria. 94. In which cell structures will the radioactivity first become concentrated? Nucleus and mitochondria. 95. Which animal cell would have the most extensive Golgi bodies? Goblet cells. 96. Which row correctly matches each cell structure with its function? Microtubules form the spindle during prophase and rough endoplasmic reticulum transports proteins. 97. Which statement supports the fact that mature plant cell can carry out the same role as lysosomes? A range of hydrolytic enzymes can be found within mature plant vacuoles. 98. A cell wall supplied with cytosine labeled with radioactive carbon. In which cell structure would radioactivity be detected first? Nucleus, cytosine is part of DNA. 99. An electron micrograph of a cell shows large quantities of rough endoplasmic reticulum and many Golgi bodies. What type of cell is being viewed? Lymphocyte. 100. Which row is correct for a typical plant cell? It has cell wall made up of cellulose, cell diameter, 5 to 40 micrometers and 70S and 80S ribosomes. 101. Where are cisternae found in a cell? ER and Golgi body. 102. What are found in chloroplast and mitochondria? DNA, 70S ribosomes, and mRNA 103. Which organelles are enclosed in single phospholipid bilayer and contain hydrolytic enzymes? Lysosomes 104. Plant cells are fixed, stained, and viewed using a student microscope. The light source was natural light. What would be clearly visible at X400 magnification? Nucleoli 105. Which size of ribosomes is found in chloroplasts? 70S 106. Which structure synthesizes and transports lipids in a typical animal cell? Smooth ER. 107. Which structure is only found in typical eukaryotic animal cells? Centriole. 108. Which parts of a cell contain ribosomes? Chloroplast, mitochondria, nucleus, and cytoplasm 109. Which types of RNA are found in both prokaryotic and eukaryotic cells? mRNA, rRNA, and tRNA 110. What is found in both chloroplasts and mitochondria? 70S ribosomes and circular DNA 111. Which organelles are required for the formation of lysosomes containing hydrolytic enzymes? Mitochondria, RR, and Golgi apparatus. Lecture notes for AS Level Biology by Muhammad Rashid. Chat the cell number 03004610455726. 112. For which cell component would nanometers, nm, be the most appropriate unit of measurement? A cell surface membrane 113. Which statements about both mitochondria and chloroplasts are correct? Both of them contain circular DNA molecules and produce ATP 114. What are correct locations of ribosomes in the typical eukaryotic cells? Free in the cytoplasm and attached to the outside of the ER 115. Which of the cell organelles will be visible under the high power, X400, of the light microscope? Chloroplasts only 116. Inside a cell, a damaged mitochondrion can be surrounded and enclosed by a membrane to form a vesicle. What happens after the fusion of a lysosome with the vesicle? Hydrolytic enzymes catalyze the breakdown of mitochondrion. 117. Lysosomes have a variety of different shapes and sizes, making them difficult to identify. What describes a lysosome? A vesicle enclosed by a single membrane, containing several different hydrolytic enzymes that may act inside or outside the cell. 118. Which order of organelles is correct and a protein is synthesized and secreted? Nucleus, ribosomes, Golgi body, vesicle 119. Which size of ribosome is found in chloroplasts and typical prokaryotic cells? 70S 120. Which structures in plant cells have double membrane? Nuclei and mitochondria 121. What are found both in mitochondria and typical prokaryotic cells? 70S ribosomes and circular DNA 122. What is the function of plasmodesmata in plant cells? To allow the symplastic movement of substances between cells 123. What is the diameter of a typical plant cell? 4.0 by 101 micrometers 124. What is the function of the nucleolus? The synthesis of rRNA. 125. Density gradient centrifuges are used to separate cell structures by their relative density. Larger cell structures have greater density and sink further down the centrifuge tube. What is the correct order of cell structures starting from the top of the centrifuge tube? Ribosomes, mitochondria, chloroplasts, nuclei. 126. Proteases are a group of enzymes that digest proteins. Which statement about proteases is correct? A bacteria protease that is secreted from disease-causing bacterium could act as an antigen and cause the production of an antibody. 127. Which sequence shows some of the stages in the production and secretion of an enzyme? Ribosome RR vesicle Golgi apparatus 128. Tay-Sachs disease occurs when cells are unable to produce an enzyme leading to a buildup of certain lipids in cells, which cell structure would not function correctly, resulting in the disease. Lysosome 129. Which part of the cell is often continuous with the RR? Nuclear envelope 130. A cell secreting glycoproteins is supplied with radioactively labeled glucose, which is detected in the cytoplasm first. In which organelle would radioactively labeled glucose be detected next? Golgi complex 131. In order to complete the sentence below, what is the correct process in cell? Structure. Cells which have a high rate of exocytosis will have many Golgi. Vesicles. 132. An amino acid enters a cell and is then used to synthesize an enzyme secreted by the cell. What is the sequence of cell structure involved in the synthesis of the enzyme? First last. Ribosome ER Golgi apparatus exocytotic vesicle 133. Which organelle does not contain a partially permeable membrane? Ribosome 134. What is the order of size of cell structures? Largest, smallest, nucleoli, lysosomes, centrioles, ribosomes 135. Which adaptation would increase active transport of carbohydrate from a plant cell? Large surface area of the cell surface membrane. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455727. 136. When mitochondria are extracted from cells for biochemical study, they are usually kept in 0.25 moles DM, 3-sucrose solution. Why is the sucrose solution used? To prevent the mitochondria form changing in structure. 137. What explains why cells with no nucleoli die? They do not have ribosomes and cannot synthesize proteins. 138. For which process is the large surface area of the cristae in the mitochondria important? For enzyme action. 139. Many single-celled animals living in fresh water possess vacuoles which contract regularly, expelling excess water. Why do the cells of plants living in fresh water not require such vacuoles? Plant cell walls limit cell size. 140. Which row shows features of a typical eukaryotic cell? Cell size, greater than 2 micrometers, nucleus, present, DNA, linear, ribosome size, large ADS 141. Which animal cell would have the most Golgi apparatus? Goblet cells 142. Which statements about a typical eukaryotic cell are correct? It has a nucleolus and linear DNA. 143. Which cell structure can be seen only with an electron microscope? Ribosome 144. What is the function of SER? Steroid synthesis. 145. Where are enzymes made? In ribosomes at RR 146. Which organelles are required for the formation of hydrolytic enzymes found in lysosomes? Mitochondria, RR, and Golgi apparatus. 147. What is the function of nucleoli? Formation of ribosomes. 148. The following are the features of eukaryotic cells. 1. Chloroplast. 2. Golgi apparatus. 3. Lysosome. 4. Mitochondrion. 5. Nucleus. Which of these has a single membrane? Golgi apparatus and lysosome. 149. Which row correctly identifies all the locations of ribosomes in a eukaryotic cell? Free in cytoplasm, in mitochondria, attached to ER, attached to nuclear envelope, and in nucleus. 150. Which structure indicates that the cell is a secretory cell? Vesicle. 151. A microscope has a resolution of 200 nanometers. Which of the following organelles would not be resolved using this microscope? Ribosomes, 18 to 22 nanometers. 152. The following are all features of eukaryotic cells. 1. Chloroplast. 2. Endoplasmic reticulum. 3. Lysosome. 4. Mitochondrion. 5. Nucleus. Which of these have a double membrane? Chloroplast, mitochondrion and nucleus. 153. ATP molecules are synthesized in mitochondria. Which sugar is found in these ATP molecules? Pentose. 154. Which statements about ATP are correct? It can be hydrolyzed to form ADP. 155. In which of these organelles is ATP synthesized? Chloroplast and mitochondrion. 156. It is suggested that primitive prokaryotic cells may be ancestors of certain organelles in eukaryotic cells. Which organelle is most similar in composition to a typical prokaryote? Mitochondria. 157. Which row shows a comparison that is not correct between a typical prokaryotic cell and a typical eukaryotic plant cell? Prokaryotic cell, all ribosomes approximately 18 nanometers in diameter. Eukaryotic plant cell, all ribosomes approximately 22 nanometers in diameter. 158. What are features of prokaryotic cells, chloroplast and mitochondria? Ribosomes. Smaller than ADS, circular DNA and surrounded by a double membrane. 159. Which range of cell diameter is typical for prokaryotic cells? 1 by 103 nanometers to 5 micrometers. 160. Which row correctly shows the presence of a cell structure in a typical plant cells and in typical prokaryotic cells? Both have 70s ribosomes. 161. 3 molecules found in prokaryotes are listed. 1. Phospholipid. 2. Protein. 3. Peptidoglycan. Which molecules are found in prokaryotic cells' surface membranes? Phospholipid and protein. 162. What are found in typical prokaryotic cells and in typical plant cells? Cell wall, circular DNA and cytoplasm. Lecture notes for AS Level Biology by Mohammed Rashid Chat. The cell number 03004610455728. 163. The DNA of typical prokaryotes is naked and circular. Which statement describes how the DNA of eukaryotes differs from the DNA of typical prokaryotes? Only DNA of eukaryotes has proteins attached to it and is linear. 164. Mitochondria have thought to have evolved from prokaryotic cells that were ingested by ancestral cells. Which feature have these prokaryotes lost during their evolution into mitochondria? Cell wall. 165. Which structures are found in typical prokaryotic cells and also in typical plant cells? Cell walls. 166. A prokaryotic cell which is 0.25 micrometers in diameter is magnified 50,000 times on an electron micrograph. How big will its diameter be in the electron micrograph? 1.25 x 101 millimeters. Image equals magnification x actual. 50,000 x 0.25 equals 12500, i.e. 12.5 millimeters. In scientific notation 1.25 x 101, 167. Which processes occur in eukaryotes and prokaryotes? Hydrolysis, transcription, and translation. 168. What is the diameter of a typical prokaryotes, e.g. Streptococcus? 7.5 by 102 nanometers. 169. Which structures are found in both typical eukaryotic cells and typical prokaryotic cells? 70s ribosomes and circular DNA. 170. Which statement is correct? Prokaryotes and chloroplasts have 70s ribosomes that are the sites for translation and polypeptide synthesis. 171. Which is correct comparison between a typical prokaryotic cell and a typical eukaryotic cell? Prokaryotes have naked DNA and eukaryotes have DNA associated with protein. 172. A culture of human cells had its cell surface membranes removed, releasing the cell contents. This material became contaminated by bacteria. The material was then centrifuged, separating out the various cell structures according to size and mass. Which cell structure would be separated out along with the bacteria? Mitochondria 173. What is correct for a typical prokaryotic cell? Cell wall has peptidoglycan, cell diameter, 1 to 5 micrometers and ribosomes 70s. 174. What is the diameter of a typical prokaryotes, e.g. Streptococcus? 7.5 by 102 nanometers. 175. Which are the characteristics of a prokaryotic cells? Absent and genetic material DNA, present 176. Which of the following are found in both eukaryotic and prokaryotic cells? Deoxyribose, lipids and ribose 177. Which structure is present in all eukaryotic cells but not present in prokaryotic cells? Chromatin 178. What are always present in prokaryotic cells? Ribosomes 179. Which statement about viruses is correct? They all have a capsid made of protein. 180. Which statement about viruses is correct? They contain DNA or RNA and use host cells to synthesize virus proteins. 181. 182. A prokaryotic cell is 5.0 micrometers in length. A virus particle is 300 nanometers in length. How many times large is the prokaryotic cell compared to the virus particle? 17. Lecture notes for AS Level Biology by Mohammed Rashid Chat. The cell number 03004610455729. 183. Which row shows a combination of molecule that could be found together in a single isolated virus? DNA and protein only 184. Which features can be present in viruses? DNA, RNA, and protein coat 185. Which statement is correct about a virus? A virus is composed of a protein coat which may surround RNA or DNA. 186. Which of the following has a single membrane in eukaryotic cells? Golgi apparatus and lysosomes. 187. What are the characteristics of a prokaryotic cell? Endoplasmic reticulum, absent, genetic material, DNA 188. Which of these are found in both eukaryotic and prokaryotic cells? Deoxyribose, lipids and ribose 189. Which order of organelle is correct and a protein is synthesized and secreted? Nucleus, ribosome, Golgi body, vesicle 190. Which structure indicates in a eukaryotic cell that it is a secretory cell? Vesicle 191. What restricts the resolution of light microscope? The wavelength of visible light. 192. Which range of sizes will include most eukaryotic cells? 1 by 101 micrometers to 1 by 102 micrometers 193. Which cell components contain mRNA? Chloroplasts, mitochondrion, nucleus and rough endoplasmic reticulum 194. Ribosomes exist as separate subunits that bind together during protein synthesis. What do these subunits consist of? RNA and protein. 195. Where would cisternae be found in a cell? Endoplasmic reticulum and Golgi. Apparatus 196. What is diameter of a typical plant cell? 4.0 by 101 micrometers 197. In general, eukaryotic cells undergo division much slower than prokaryotic cells. What is the reason for this? Eukaryotes break down the nuclear membrane during mitosis. 198. Which cell components are present in all prokaryotic cells? Cell wall and cell surface membrane. 199. Where would Christie be found in a cell? Mitochondrion 200. What is diameter of typical prokaryotic cell such as Streptococcus? 7.5 by 102 nanometers 201. A human aorta has a lumen width of 2 centimeters. A human red blood cell has a diameter of 7 micrometers. How many red blood cells could be laid end to end across the diameter of aorta lumen? 2.9 by 103 202. Which of the structures are found in photosynthetic prokaryotes? Cell surface membrane, mesosome and ribosomes 203. Membranous sacs containing products of metabolism are formed by the endoplasmic reticulum in cells. Where are these products used? Inside and outside the cell. 204. Using a stage micrometer scale, 1 unit of eyepiece graticule was calculated as 0.005 mm. The diameter of the spongy mesophyll cell was counted as 3.5 units on the eyepiece graticule. What is the estimate diameter of the cell? 18.0 205. Which type of cell will contain the highest proportion of single membrane-bound structures? Goblet cell 206. Which structures are present in a typical plant cell? Mitochondria and vacuole 207. Which the correct comparison between light and electron microscopes? Light microscope has low resolution and low magnification, whereas electron microscope has high resolution and high magnification. 208. A cell organelle measures for x 10 to 1 mm in diameter. What is the diameter in 4 by 102 209. Which cell organelle does not contain nucleic acid? Golgi apparatus 210. The graticule and stage are used to measure cell. Which is the correct reason why the graticule calibrated? The graticule can be used to make measurements. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457030. 211. Which of the cell organelles, when appropriately stained, will be clearly visible under the high-power X400 of light microscope? Chloroplasts only 212. Which structures are measured using these units? 10 to 3 m, xylem vessel, 10 to 6 m, nucleus, 10 to 9 m, ribosome 213. What is the function of smooth ER? Steroid synthesis 214. A lymphocyte has diameter of 1x 10 to 2 mm. What is the diameter in nm? 1x 104 215. Which cell structure has been seen only with an electron microscope? Cell surface membrane 216. A plan diagram is made of a transfer section of a leaf. Which features would be seen in the diagram? The overall distribution of tissues and the relative thickness of the tissue layers. 217. What describes the features of an electron microscope? Maximum magnification, 2.5 by 105, resolution/nm, 5.0 x 10 to 1, specimen, dead 218. Cells which do not have nucleoli die because they do not have ribosome and cannot synthesize protein. 219. What are the appropriate units for measuring diameters of alveoli, white blood cells, and the width of cell walls? Diameters of alveoli, diameter of white blood cells, width of cell walls, nm 220. Which combination is found in a prokaryotic cell? DNA and RNA 221. What is the shortest distance traveled by an oxygen molecule diffusing from the alveoli air space into one of the red blood cells? 222. Mitochondria are thought to have evolved from prokaryotic cells that were ingested by ancestor cell. Which feature have the prokaryotes lost during their evolution into mitochondria? Cell wall 223. When mucus is secreted from a goblet cell in the trachea, these events take place. What is the sequence? Addition of carbohydrates to proteins, separation of a vesicle from the Golgi apparatus, fusion of the vesicle with the plasma membrane, secretion of a glycoprotein. 224. What is the order of size of cell components from largest to smallest? Mitochondria, nucleoli, lysosomes and centrioles. 225. A specimen is viewed under a microscope using green light with the wavelength of 510 nanometers. If the same specimen is viewed under the same conditions, but using red light with the wavelength of 650 nanometers instead, what effect will this have on the magnification and on the resolution of the microscope? Magnification remains the same, and resolution is decreased because of increased wavelength of the light from 510 nanometers to 650 nanometers. 226. In 1985 a giant bacterium Epilopicium fischelsoni was discovered. Which cell structures would be present in Epilopicium enabling biologists to classify this organism as prokaryotic? Circular DNA lying free in the cytoplasm. 227. The diameter of living cells varies considerably. Typical diameters are, a prokaryote, such as Streptococcus equals 750 nanometers, a eukaryotic cell, such as a white blood cell equals 15 given the diameter of the white blood cell is how many times greater than the prokaryote? X 2228. What is the function of RR? Protein synthesis and transport. 229. In which animal cell would Golgi apparatus would be most abundance? Goblet cells. 230. What is the resolution in nm of an electron microscope and a light microscope? Electron microscope, 0.5, light microscope, 200-231. An amino acid enters a cell and is used to synthesize an enzyme secreted by the cell. What is the sequence of cell components involved in this pathway? Ribosome Golgi apparatus exocytotic vesicle 232. The action of which cell depends on large number of lysosomes? Phagocytes 233. What is the correct order of size of cell organelles from the largest to the smallest? Nucleus, mitochondrion, lysosome, ribosome 234. Which structures are found in plant cells but not in animal cells? Plasmodesmata. Lecture notes for AS Level Biology by Mohammed Rashid Chat. The cell number 03004610455731.